Yes.
Okay, we did till the monocystronic and polycystronic arrangement of the gene. Very well. So I think where did we start in the last class? The cellular machinery, right? We started from the cellular machinery. Okay, cool. So who will tell me about the cellular machinery quickly? Anyone who would like to volunteer, or should I pick? Please, someone should volunteer, right? These are interesting things. Anyways, when I will pick you, people will like, you will do good. You will explain it well. So why not explain it on your own? Atif, Fatima, Rifat, Hiba. I think Rifat is not able to speak. Rifat, please fix your mic because you are losing on to some important discussions. I'm not hearing from you. You're just answering in yes, no's and things. So I'm not understanding your growth in the class as a student. Okay, unless and until you will speak, I'll not know how much you are growing. I know about Atif, Fatima, and Hiba to some extent. Okay, Hiba, are you there? Can you speak today? Hiba, can you speak today? Can you give the summary today? The last class? No? If you cannot speak, you can write. Okay. I'm always left with Atif and Fatima then. Fatima, how about you? Yes, no, something. No one will speak today? Yes, Rifat, please get it fixed because, otherwise it's difficult. The way I conduct classes, it has to be discussive because the only time I enjoy teaching is when there is some cross questions, there are some inputs from the students asking, advising, suggesting, etc. Fatima also cannot speak today. Okay, then Atif, what about you? It's you and me then.
Yes, sir. Can you hear me?
Yes, I can hear you. I was saying it's you and me then. So I will do the teaching. So can you summarize last class?
Yes, sir. I'll try, sir.
Yes, very good. Thanks.
Yes, so we learned about the cellular machinery of DNA replication. We learned that the main enzyme responsible for replication is DNA-dependent DNA polymerase. But DNA-dependent DNA polymerase can only synthesize from 5' to 3' direction. So due to that, when it enters the DNA, in one strand, it forms a continuous strand, but the other strand is discontinuous and it's like fragmented. You also learn that DNA polymerase has a very high rate of synthesizing DNA strands. It can synthesize about 2000 base pairs per second.
Yes.
Then, yeah, we learned that continuous strands are also known as leading strands, and discontinuous strands are also known as lagging strands. So then lagging strands are later joined by the enzyme known as DNA ligase.
Yes, and in that lagging strand, the fragments which are synthesized are also known as Okazaki fragments, which are basically joined together. Okay.
Yes, sir.
Yep. Okay, very well. So if they ask you to make a diagram of the replication fork and explain the process, you'll be able to do it, right?
Yes, sir.
So this is whole, everything that you have made and marked here is the replication fork. Yes, Atif. Then we went on to the rate of replication. You there, Atif?
Yes, sir. Yeah, so we calculated the rate of replication through the cell reproduction process of E. coli, and we found out that DNA synthesizes base pairs of about 2,000 of them per second. Due to
Yes.
the speed of this process, we also learned that this very expensive when it comes to energy.
Yeah, and it's also very error prone. Okay, it is energy expensive process. It is also error prone. Okay.
Yes.
Yeah.
Sir. Then we learned about transcription, which is the process of copying genetic information from a strand of DNA into an mRNA. We learned that only one strand should be used to make mRNA. This is because of two reasons. That is, one is if both of the strands are acting as a template, then two different mRNAs will be coded, which will lead to translation of two different proteins, which is not desirable. Then there is also a chance that these two mRNA are complementary to each other, then they will bind to form a double-stranded RNA, which cannot be understood by ribosomes and therefore cannot be translated.
Yes, right. And then what was transcription unit?
Yeah, we learned about the transcription unit, which is like the part of a gene which is used for transcription. We learned that there is three parts: the promoter, which is like the starting part, then the structural gene itself, then the terminator, which is like the end part. So we also learned the enzyme responsible for the transcription is DNA-dependent RNA polymerase, which also catalyzes the polymerization in 5' to T' direction, similar to that of DNA-dependent DNA polymerase.
Yes.
So due to this, the strand with the polarity 3' to 5' will act as a template due to the because DNA is anti-parallel. So the other DNA sequence has the same sequence of the mRNA produced. So therefore, this strand is known as the coding strand.
Yeah, right. Though it does not code for anything, right? It is not participating in that whole process. But
Yes.
Still it's named coding strand because I gave you the reason. Probably it's because the coding sequence of the mRNA, which is produced from the template strand, is same as of the sequence of the coding strand. Yeah.
Yes. Then we
And
learned about the transcription unit.
Just
Yes,
one
sir,
question. Yeah, Atif. I was asking how to figure out the transcription direction. By looking at the promoter and the terminator. Okay. So the direction of transcription is always from promoter towards terminator. So that the DNA dependent RNA polymerase will sit here on the before the promoter, and then will start synthesizing the mRNA from the structural gene. And the moment it will reach the terminator, it will fall off. So transcription will be over. Okay. If they ask you, how do we figure out the direction of transcription? Okay, so it's figured out from the promoter towards the terminator. Okay, or
Yes,
you know the direction of the DNA dependent RNA polymerase 5' to 3'. So you can say the direction of the transcription will be 3' to 5' of the template strand, right?
Yes, sir.
Yeah, multiple ways. Okay, continue. Then we come to the last part.
Yeah, okay. Then we learned about the transcription unit and genes. We learned that generally DNA is transcripted to form RNA, but there are many types of RNA, that is mRNA, tRNA, and so on. So since we have to classify them, we classified the segments of the DNA which code for proteins as cistrons. And then we learned that transcription units may be either monocistronic, that is for eukaryotes, or polycistronic, that is for prokaryotes.
Monocistronic means?
Monocistronic means
monocistronic. You already know what cistron means. You already know what mono means. You can look at the board also. Monocistronic means the the gene which is present between the promoter and the terminator, it codes for only one protein. Okay.
Yes, sir. I got it. You got confused.
Yeah. So mono means single, cistronic means a segment of the DNA that codes for a polypeptide chain, right? So monocistronic is the segment of the DNA that codes for only one polypeptide chain. Whereas polycistronic is where the segment, one segment present between the promoter and the terminator can code for many polypeptide chains. This happens in prokaryotes, like in bacteria. Okay. Yeah. Then explain this. What did we draw here? What is this?
Yes, sir. So we learned in eukaryotes that monocistronic structural genes have interrupted coding sequence. That is, the genes in eukaryotes are split. So yes. So in the DNA, the structural gene is divided into introns and extrons. Introns and exons. When the DNA is transcribed into mRNA, the final mature mRNA only contains the exons. The introns are removed.
Yes, right. And that process of removal of introns, write down. Now from here I start the class. So just add the first thing of today. The process of removal of introns from an mRNA to make it a mature mRNA. Is called splicing. Splicing, exactly like you splice something. Here the mRNA is being spliced. It's called splicing. Okay.
So don't doubt.
Yes, please.
Yes,
How are they joined?
Do introns and exons have any like structural difference, or is it just classified on the basis that the final mature mRNA does not have it?
Yeah, the difference is in the coding. So introns, if you retain introns in the final mRNA, so when you will make the polypeptide chain, the part which intron will code for will not be a part of the protein. So it will be some extra garbage which will be now become part of the protein. So the protein will be non-functional. So in order to make a complete protein, a functional protein, introns has to be removed before the ribosomes come and start reading the mRNA to produce the protein. This splicing has to be done. And this is known as post-transcriptional modification. What is called? Post-transcriptional. So it's happening after transcription, right?
Yes.
It's called post-transcriptional modification. And the process is called splicing, and the introns are all removed. The exons are all present. Now, one question could be, when you remove, basically you are creating a cut, right? You are chopping it, you are splicing it. So how is the mRNA again joined back? You already know the answer. How do we join back nucleic acids? How are we joining the Okazaki fragments?
DNA ligase.
Yes. So what will be present here?
RNA ligase.
Yes, perfect. Okay. So they are joined back to form a mature mRNA, which goes to the ribosome, and then the protein synthesis takes place. Is it clear?
Yes, sir.
Yep. Okay. So the difference is that this sequence, there is no like structural, because it is the sequence. The structure of the mRNA is the sequence continued. So it is like some sequences which are not required for the protein. Yes, Rifat, I'll explain splicing again. So splicing is where I told you a monocistronic gene is arranged in a split manner into introns and exons. Okay. So after the promoter and before the terminator, this structural gene has exons and introns, one after the other. Exons are the important regions which actually codes for the protein. Introns are removed through splicing. Okay, it is called post-transcriptional modification. Okay, one of the post-transcriptional modification. There are other modifications about which we will read today. So when you remove these introns, you are basically chopping the DNA at an exon and intron junction and removing all the introns, degrading it and joining back the exons. Okay. So this way, the final mRNA which is produced is only containing exons and introns are removed. This process is called splicing. The process of removal of introns from an mRNA to make it a mature mRNA ready for translation is called splicing. Clear, Rifat? You understand? Say yes or no. Yeah. Okay, very well. So today we'll be talking about the various, the different types of RNA and which play a role in the process of transcription or which come through the process of transcription. So write down types of mRNA, not mRNA, types of RNA. mRNA is one type of RNA. Types of RNA and transcription process. Okay. Now, as I told you, there are three types of major mRNA. Okay. So write down. There are three major types of RNA. First one is known as mRNA, about which we have studied the most till now. mRNA is known as, like it's called mRNA because it's messenger mRNA. Messenger RNA. This is what contains the information to form protein. This is the only mRNA that actually forms the protein, like has the message to form the protein. The other two help in the process, but they are not directly read, but they help in reading and bringing material. So the second one is tRNA. The name here is transfer RNA. Okay. Now you know that it transfers something. Now what it transfers we'll come to know now. And the third one is rRNA. And this is called ribosomal RNA. Make sense, people? Messenger RNA, transfer RNA, and ribosomal RNA. Okay. Now all these three RNAs are actually needed or are required to synthesize protein. But the one RNA which actually contains the information which protein will be synthesized is the mRNA. Okay. So what is required to make a protein? Tell me. How do we make a protein? You know about ribosome. You have studied in junior classes. What is ribosome? Quickly. What is ribosome? Atif? Fatima?
Ribosomes are organelles in the cell that help for protein synthesis.
Yes, but I'm talking about the structure. Any idea about what are they? Because they are non-membranous. This you know, right? That's why they are present both in bacteria and eukaryotic cells. So they are present both in prokaryotes and eukaryotes, correct? But is the ribosome present in the prokaryote same as the ribosome present in the eukaryote? What do you think? Do bacteria and humans have the same ribosomes, same kind of or type of ribosomes in their cells?
No.
Forgot everything you studied in. Yes. So what is the difference? Prokaryotic ribosome is called 70S ribosomes, right? And eukaryotic ribosome is 80S ribosome. S is the Swedberg's constant, the sedimentation constant. Remember? Even their subunits are different. They have two subunits, bigger and smaller. Right?
Yes.
So the ribosome is different, but what is ribosome made up of? If it does not have a membrane, what does it have? You know what is a ribosome made up of? A ribosome is made up of RNA and protein. So there is a RNA. Okay. Because it's RNA, it's single stranded. And then there are protein structures, beta pleated sheets, and just like this. So this is the organelle. So the green part here is protein. And the red part here is RNA. And this RNA will be known as ribosomal RNA. You understand? And together, this is the ribosome. Now can you study how can it read? Now can you understand how can it read a mRNA? Because mRNA is also single stranded. Yes or no? That mRNA, this rRNA helps in reading this mRNA. Both are single stranded, so it can help in reading the bases. Yes or no?
Yes.
And there will be a tRNA. So this tRNA, what is the other thing? So one thing we need is a ribosome, which has a RNA in the core, and it is, so basically a ribosome is nothing but a mRNA and a protein structure. No membrane, nothing. That's why it's present in both eukaryotes and prokaryotes. That's why it's a very different cell organelle. So one thing is we need ribosome. The other thing to make protein, we need mRNA. What is the other thing? What is the most important ingredient to make protein? What is protein made up of? What is protein made up of? People. Nutrition chapter class 11th. When we break down proteins, what do they get broken down into? Protein digestion. You really need to brush up some concepts, okay? All of you. If you don't know this, it's bad. I expect you to know these things. If you cannot speak, you can write in the chat, but it's difficult for me to move forward without you knowing these basics. When we eat protein, there are peptidases in our body. Remember trypsin, pepsin, chymotrypsin. They break down protein in the small intestine and they convert the protein into what? What is the monomer of protein? Someone has to give me this answer. Go to Google, go to your 11th class textbook right now. If you just, but tell me, what are proteins made up of? You all know it, actually. Atif, Fatima, Rifat. What are proteins made up of?
Amino acids.
Exactly. It took you so much time to figure it out. Googled it or what? We break down all the proteins into amino acids in our body, right? And then the blood absorbs it. If you are going to higher classes, do not forget your junior classes. They are the foundation. You know, if you wish to clear NEET, you have to know the foundations, right? So
Yes.
amino acids make proteins. So if ribosome can read this messenger RNA to make a protein, protein is known as a polypeptide chain. I have just written here. Polypeptide chain. So what is this polypeptide chain? It is So this is how the, now let me make a simplified version of this. There is a bigger subunit and there is a smaller subunit of the ribosome, right? And let's say this is the blue one is the mRNA. So the mRNA is red. It goes inside from one side and comes out. So this is the mRNA. Understand everyone?
Yes.
Inside this ribosome will be this rRNA, right? Ribosome,
Yes.
mRNA. So rRNA is present. Sorry. rRNA is involved through ribosome. mRNA directly is red. And then there is something which I'm making here in green. Let me use some other color, make purple. So there's something which is called tRNA. tRNA is like this kind of a RNA structure. It's like clove shaped. So this tRNA brings an amino acid, any amino acid with it. So one tRNA is carrying one amino acid. How many amino acids are there in human body? How many amino acids takes part in the protein synthesis? There are 20 amino acids, 22 in human body total. Like we studied 20, but there are 2 more discovered. The 2 more are actually some modified parts of the earlier existing ones. So there are 20 different amino acids. Okay, so all these 20 are loaded on one or the other tRNA. These tRNA bring the amino acid to the site where it is being added. So this is how one after the other amino acid is kept like joining one at the top of the other. You understand? So this will move forward, then the new one will come and join here. Then this whole chain will move forward, then next one will, and mRNA will also keep moving forward. This will also keep moving forward. Do you understand?
Yes.
And tRNAs will keep coming, and this is a tRNA. So there'll be many tRNA, hundreds of tRNA carrying all the amino acids. One by one, they will keep coming and keep depositing the required amino acid. And how is it decided which amino acid will come? Because here there is a 3 base pair codon which is red, right?
Yes.
So on the basis of the codon, it is decided that which amino acid should come, and that comes and gets added. And this is how the protein synthesis happens. So can you see now mRNA, tRNA, and rRNA all plays a role in protein synthesis, right?
Yes.
Yes. So write down. The mRNA provides the template. The mRNA provides the template. The tRNA brings amino acid and read the genetic code. Brings amino acid and read the genetic code which is here and then deposit it accordingly. And the rRNA plays structural, because it forms the structure of the ribosome, plays structural and catalytic role. It catalyzes this whole process. You know what are catalysis? Catalysts are the chemicals which speed up the process, right? With less energy required. You know about catalysts, right? From chemistry. chemical catalysts?
Yes.
And I already told you that RNA has this property of behaving like a catalyst, right? That's why the RNA world hypothesis came. So is everything making a connection in your head, and is everything getting cleared now?
Yes.
Yes. So these three mRNAs are required for protein synthesis. All right, down. All the three type of RNAs. All the three type of RNAs in sorry, write down in prokaryotes. So these three type of RNAs are present both in prokaryotes and eukaryotes. Okay. So in prokaryotes like bacteria, all the three type of RNAs are synthesized or transcribed. All the three types of RNAs are transcribed through a single DNA dependent RNA polymerase. Is it clear? So there is only one type of RNA polymerase in the bacteria which transcribes all the three types of RNAs: tRNA, mRNA, and rRNA. Okay. So transcription is different in prokaryotes, slightly different in prokaryote from the eukaryotes. Now, one reason is that there is only one type of RNA polymerase. Second reason is the prokaryotic mRNA does not needs post-transcriptional modification. No splicing is done there because exons and introns are not present in prokaryotes. Is it clear, everyone?
Yes.
Please write down this information, draw everything which is on the board and let me know, then I'll move forward. Now I'm going to tell you about the difference between bacterial transcription and eukaryotic transcription after this. Everything which is on the board is clear? If you have any doubt, please ask. Do not allow me to move forward with a doubt in your head, because that will create problem further on. So when the very crucial parts of any concept comes, I move a little slow for your sake. So let me know if you find any difficulty. This is polypeptide chain. Done, people. Shall we go to the actual process, the complicated part? Yes, Rifat is done. Others? Everyone is done? Fatima, please write down if you cannot speak. At least you can write in the chat, I believe. Don't be totally silent. Okay. If you are making, shall I move?
Yes, sir, I just finished.
You done?
Yes.
Okay. So let's write down transcription in prokaryotes. First. In prokaryotes. Now it is simple because I told you there is only one RNA polymerase, which is DNA dependent. It transcribes all the three type of RNAs, tRNA, mRNA, rRNA. And it also starts the transcription, makes the mRNA, and then ends the transcription. But for that, it needs some help. Now, the first process that happens is, let me make a DNA here. Say, this is the DNA, okay? Okay. Okay, everyone. Now, the first step will be to bind. So this RNA polymerase, so this is the DNA. Let's say there is a gene, the promoter for that gene starts from here. So this is the promoter region. Okay. So this is the promoter region. If this is the promoter region here, and let's say this is the DNA dependent RNA polymerase. Okay. So this has to come and bind at the promoter region, correct? Also, let's write the polarity. And let's say this is 3' to this 5'. So it goes like this. This is 3', this is 5'. So which strand will it bind to? RNA polymerase will bind to which strand, which will be the coding template strand. The direction of synthesis for the RNA poly is 5 to 3. So this 3 to 5 will be the strand with which it will bind, right? But before first, it has to recognize the promoter sequence and has to bind here. So I'm just making this strand. Can you see this region that I have marked? I am making it yellow. Okay. This region on this 3', 5' strand is the promoter region. Now to bind here, there is one factor that helps. Without this factor, you cannot bind, and that factor is called a sigma factor. So this sigma factor comes and binds to this DNA dependent DNA polymerase. This is sigma factor. And this sigma factor tells the DNA, the RNA polymerase to bind at the promoter. So this initiates the binding. So this is the first process. The first stage is initiation. initiation of transcription. So in that initiation, the RNA polymerase gets some help from a factor called sigma factor, which binds on the RNA polymerase and tells it's like a guide. It binds, holds the finger and tells the RNA polymerase, okay, come, sit here. This is the promoter region. You understood? Everyone?
Yes.
So we can write its name. It's called sigma factor. Okay. Sigma factor, promoter, RNA polymerase, done. Initiation is done. Now, the second step will be elongation. Okay, now for the elongation, what happens is, we go a little down. So let me again make the DNA. This time it started, let's say, from here. So it's a little tricky to make because this is a good way to make it. I hope you understood what's happening here. The DNA opened, right? Can you see this is opened from here?
Yes.
So just after the promoter region. So here was the promoter region, right? This. The DNA opens up. And to keep doing the transcription, the polymerase has to bind with the 3' to 5' strand. So let's say this one here, how is it going? This is 3' to 5' strand. So the factor, the RNA polymerase goes and binds to this strand. Okay, clear? So this same factor went inside now. And once it starts elongating, it starts moving, it is making a trail of this mRNA. So what you see here is the mRNA, okay? This single stranded thing is the mRNA. Clear, everyone? And now the sigma factor actually is out. It's no longer bound to the polymerase. Is it clear?
Yes.
RNA pole, and this is the RNA. And this process, so where was the terminator, where was the promoter gene? It was here, let's say. This was the region where the promoter gene was. And from now on, from promoter gene onwards is now the structural gene, right? And somewhere at the end will be a terminator region. You understand? So this process is called elongation because the mRNA is being formed. The mRNA is being elongated. You understood elongation? And let me use one different color for, let's use brown. Somewhere here is the terminator region. And this was promoter region. Now you understand between promoter and terminator, all of this is the, what is all of this? All of this is
Structural gene.
Yes, structural gene. And this mRNA is coming, which is coming, it is basically the structural gene part, right? So it will keep going forward and it will keep making the mRNA. This is understood, everyone?
Yes, sir.
This is the second process. Now, after elongation, it has to terminate, right? So now when it will reach, let's say this side, so that there. So only a small part of the DNA, as you can see, remains open. As the RNA keeps moving forward, the part which it has surpassed again becomes paired and wound. Okay. Now the polymerase reached here. Okay. So when it reaches here, there is something that again has to give it a signal to stop, because here is the termination region, right? This region is the terminator region, correct? So when it reaches here, there is a factor. Again, it needs some help, and comes a factor which is known as Rho factor. So I'll make a Rho factor, let's say alpha, and make it dark green. So there is another factor that binds to it. It's called Rho factor. Okay. Now this Rho factor is the terminator factor. This is the initiation factor. This is the termination factor, and it basically makes the mRNA leave the DNA and come out, fall off, and the make the polymerase leave the DNA and fall off, and as a result, the mRNA which was formed also falls off. Okay. Understood, everyone?
Yes, sir.
So RNA polymerase falls off upon binding to termination factor, the Rho factor. So here also. Okay, is it clear, everyone?
Yes, sir.
So here, somewhere here was the promoter region. So it reaches the termination region and falls off. And all this gene, this structural gene part, got transcribed. Not use this symbol. Into this mRNA. You understand?
Yes.
Can make it a little longer. Yes. This structural gene got transcribed into this RNA, mRNA. Correct? Now, this mRNA does not require any post-processing because there is no split gene arrangement here. Here, it's polycistronic. So between one promoter and one terminator region, there will be multiple polynucleotides formed. So the bacteria does not care, does not worry about anything. And you already know that in bacteria, there is no specific nuclear chamber and a cell cytoplasm, right? It is not well defined, right? The DNA is just floating around in a dense region called nucleoid in the cytoplasm only, correct? So the moment the mRNA comes out, it starts getting translated. into polypeptide chain. Sometimes not sometimes, but what happens is when the mRNA was just coming out, it was already, the ribosomes were already come here, and the mRNA is not yet fully formed, and the protein synthesis will start. Okay. Now this thing is very unique in bacteria and is known as the coupling. So basically the translation and the transcription is coupled here. You understand? So it's called transcription, coupled transcription and translation that happens in prokaryotes. Is it clear? In prokaryotes, it's simple. No more.
Yes, sir.
Thing. First draw this, everyone. Have you drawn this initiation elongation part? Let me know when you're done. Also, let me know if I have to scroll. Okay, so this part is, let me complete. Oh, sorry. So this part is called termination. There are three distinct things: initiation, elongation, and termination. And all these three happens with the help of the same RNA polymerase. Please make this part, then I'll scroll down. Let me know when I have to scroll down. Is the process clear to everyone? No doubts?
Yes, sir.
Yes, no? Yeah, good. If it is done, do you want me to scroll down, people? At least this initiation part is done, right? So we can just go to the termination part, elongation and termination. Yes. Done, people? Everyone done? The prokaryotic part? Yes, no, something?
1 minute, sir.
Yep. Okay, Rifat is done. So let me first dictate you about the prokaryotic part. Then we'll go to the eukaryotic part. So write down. In prokaryotes, there is a single RNA polymerase. There is a single RNA polymerase that polymerizes the mRNA. That polymerizes the mRNA, or that polymerizes all the RNAs, mRNA, tRNA, rRNA, everything. In a template dependent fashion. So it only depends on the 3' to 5' template dependent fashion. Next point, to initiate the transcription, the RNA polymerase transiently means for some time, transiently binds or associates with the sigma factor, also known as the initiation factor. Transiently binds with the sigma factor. In the brackets, you can write initiation factor. Which helps the RNA polymerase bind to the promoter region. Okay, done. Next, during elongation, the enzyme opens the helix. The enzyme, RNA polymerase, opens up the helix, the double helix, the DNA. Opens up the double helix, and only a short stretch of RNA, remains bound to the RNA polymerase. And only a short stretch of the RNA remains bound to the RNA polymerase. Okay, this is for elongation. Now, third point, when the RNA polymerase reaches the terminator region, it binds transiently with the Rho factor, in the bracket termination factor. It binds transiently with the Rho factor or terminator factor, and results in the termination of the transcription process, and results in the termination of the transcription process. And am I audible clearly, everyone?
Yes.
Yeah, results in the termination of the transcription process. And last point, people, some network issue, I just got logged off. So I'll just keep my video off for some time. Let's finish. Let's go with the audio only. Let's finish that part. Okay. Can you see the screen, everyone? Yes.
Yes.
Yes. What was the last line that you wrote? Please tell me. What was the last line?
When the RNA polymerases reaches the termination position, it binds transiently to the pro factor. Yeah, that one.
Okay. So you have not written this. The RNA does not require any processing to become mature.
No, so in the middle of the class, I left because I thought something was wrong with my internet.
Okay. Okay. So write down last point, which is the fourth point. The RNA, or it is called the nascent RNA. Why it is called nascent? Can you tell me? Nascent means the new. So this is known as the nascent RNA, which is newly formed. So you can write down the nascent RNA does not require, the nascent RNA does not require any processing to become mature. Any processing to become mature. And also there is no separation between the cytoplasm and the nucleus in the bacteria. And also there is no separation between cytoplasm and nucleus in the bacteria. Therefore, translation can begin. Translation can begin even before the mRNA is fully transcribed. Translation can begin.
Can you repeat the point?
Yes. So translation can begin even before the mRNA is fully transcribed. You wanted me to repeat this point?
No, sir.
It's okay. Written? I hope I'm audible clearly, right?
Yes, sir, I'm right.
Because you are not audible to me clearly. Your voice is breaking in between others. Is my voice also breaking?
No, sir.
Okay. So this process is called transcription coupled translation. Transcription coupled translation. So both happens simultaneously in prokaryotes. and because of two reasons. One, no processing, no post processing. Of RNA required, and second, no compartmentalization. There is no different space for nucleus and different space for cytoplasm. Both are same, and no processing is required. So even before the whole mRNA comes out from the RNA polymerase, it starts getting translated. Okay, so the protein synthesis is coupled with the mRNA synthesis in prokaryotes. Is it clear, everyone? Yes, no? Okay, so now let's come to more complicated part. Write down transcription in eukaryotes. Now in eukaryotes there are two complexities, two additional complexities associated. Can anyone tell me what are the two complexities? Any idea? No? Okay, so write down. First of all, write down. There are 3 RNA polymerases. 3 RNA polymerases in eukaryotes. Okay. All are DNA dependent, but there are 3 RNA polymerases in eukaryotes. The first one is called RNA Pol 1, RNA polymerase 1. And this forms rRNA. What is rRNA? Ribosomal RNA. Okay. Yes. And then there is RNA polymerase 2. It's called RNA Pol 2 or RNA polymerase 2. I'm writing full. This codes for there's something called heterogeneous nuclear RNA, which is not in your syllabus much, but it is called hnRNA. hnRNA, okay. If you want to write it full, I can just write it here. Heterogeneous nuclear RNA. Okay, produced for to be functional in the nucleus. And third one is the most active. This is the major in eukaryotes. Make a star. This is the hero. RNA Pol 3. RNA polymerase the third. And this codes for tRNA, tRNA, snRNA. tRNA is transfer RNA, okay. snRNA. snRNA stands for small nuclear RNA. Small nuclear RNA. and also the srRNA. So you just have to remember, I'm telling you which one, I'm marking it, which has been asked. This is asked. This is asked because this is important. I'm telling you why. This is asked. Others you can. So this is rRNA, this is tRNA. And this hnRNA actually is the precursor of mRNA. Okay. So that's why this is important. Why it is called precursor? Because in eukaryotes, you cannot produce a mRNA before you process it. So basically this hnRNA later becomes mRNA. So you understood tRNA, mRNA, rRNA. All three are coded by different all three are transcribed by different RNA polymerases. One for rRNA, two for mRNA, not directly, but three for tRNA. Okay, everyone, will you remember this? Yes.
Yes.
Yeah. So this is the first complexity, that there are many mRNA, RNA polymerases. The second complexity in eukaryotic system is that the first mRNA that comes out, which is this hnRNA, heterogeneous nuclear RNA, it actually is non-functional. Okay, because it contains both exons and introns. Write down the primary mRNA.
So what is srRNA?
Small SR is it's basically a ribosomal RNA, but it is 5S. 5S. So the full name is 5S RNA. So 5S ribosomal RNA. It's very small ribosomal RNA. Okay. So I did not use the word 5. You can just remember it as small ribosomal RNA. Now the primary mRNA, which is hnRNA, till it is in the nucleus, because we have a separate nucleus in eukaryotes and a separate cytoplasm. So till it remains in the nucleus, it is called hnRNA, and it contains both exons and introns, right? And hence this is non-functional. So this RNA is non-functional. It cannot help in protein synthesis. So what has to be done? So this hnRNA undergoes post-transcriptional modification. So write down hnRNA undergoes post-transcriptional modification to become mRNA. Okay. You understand? Now, in this post-transcriptional modification, there are three things. So I'm writing it here. The post-transcriptional modification. First thing you know already. What is it? Splicing. What happens in splicing? What happens in splicing, people?
Introns are removed.
Yes, and exons are joined. That also you have to say. Second is, once you splice it, the mRNA, when it comes out of the nucleus into the cytoplasm, it is susceptible to damage like degradation. So literally the mRNA has to be put a cap and a tail. So it's called capping and tailing. Like जूते पहना रहे हैं और टोपी पहना रहे हैं. You're putting the boots, the cap on. You understood, everyone? These are for protection of the stability of the mRNA in the cytoplasm. Do you understand? And it specifically happens in eukaryotes only, not in prokaryotes. Because in prokaryotes, before the RNA is exposed to any other thing and can get damaged or degraded, right away you do the translation. You start the translation. And there is no exon intron system. So it does not require splicing. You don't have to worry about capping tailing. Okay. So these three are post-transcriptional modifications. So these are the two complexities. So now let's start understanding with these complexities how it happens. Understood the complexities?
Sir, so
Yes,
the
no, something. Yeah.
so the ribosomes
So
translated into HRNA and later is conformed into mRNA.
Come again. Come again.
Sir, it's breaking. I'll type in the chat.
Say again. Yeah, no, I can hear you. I can hear you.
Okay, sir.
I just
So
could
the
not understand.
the ribosomes transcribe into HRNA and then later on it is converted into mRNA to modification.
Yes, right. So the first mRNA which comes out from the RNA polymerase is called hnRNA, heterogeneous nuclear RNA, because all of this process in eukaryote is happening in the nucleus, right? And nucleus is well defined through a double membrane. So it is happening in a different space altogether in the nuclear membrane, inside the nuclear membrane. So it is called hnRNA. So that hnRNA has to undergo splicing, capping, and tailing to become a mRNA. You understood?
Yes.
Yep. You're right. Now, let me make you understand how it happens. So let's make a DNA strand. Let's say so this is the DNA where in eukaryotes where transcription is happening. Okay. So it is open at this region. And let's say the RNA polymerase has already synthesized the mRNA. Now the mRNA that I'll be making will be of two different colors here. So you all are understanding?
Yes.
Yeah. So this is the mRNA where the yellow region is the intron and the purple region is the exon. Okay. This is RNA Pol. Which one is it? RNA Pol 2, right? In this case, it's RNA Pol 2. And this is 3' to 5', 3' to 5' DNA. So when this comes out from this RNA Pol 2, this mRNA, the first thing that happens with this mRNA is capping. Okay, so let me draw this mRNA separately here. It's clear, no?
Yes.
So the first thing that happens is cap. So because the first thing that comes out, the first bit of the mRNA that comes out, it requires a cap. So there is a cap which is put. Now this cap is called MG trimethyl guanosine, methyl guanosine triphosphate. So it's called methyl guanosine triphosphate, MG/P. This is the cap. Methyl guanosine triphosphate. And this process is called capping to make the head of the mRNA stable, the first post-transcriptional modification. The second thing that happens to the mRNA is a tailing. Now in the tail part, what happens is this is tailing. In the tailing, polyadenylation happens. The word poly means many, adenylation means addition of adenine. Okay, so this is a poly A tail. So there will be A, this like this. You understood? Multiple A's are added at the end. G is added at the capping, A is added at the end. Now this mRNA is stable. Only thing it needs now is splicing, right? But still
Yes,
it
sir.
is hnRNA. So this part was hnRNA. When this capping and tailing happens, it now needs to be spliced. The splicing needs to happen. So what will be removed? The introns, the yellow part will be removed, and you will see the mRNA will look like this. It will be short with a poly A tail and with a cap, right? And you will see that these yellow regions will be spliced and removed, right? These were the introns, right? So introns are spliced and removed. And only the exon is left. Ligated. So this now becomes a mRNA. Mature RNA, ready to go into the, where?
Ribosome.
Yes, but where will it go? It will now go into the cytoplasm. Yes. And in the cytoplasm, it will bind with ribosomes. And what will happen? Translation will start. Is it clear? Everyone, is it clear? So this is the messenger mRNA. Please draw this. Remember that it is RNA Pol 2. Yes. And one more thing, this poly A tail. So this poly A, it's around 200 to 300 adenines. It's not just 10, 20. Let me know when you're done, people. To draw this, I'll be back in a minute. Okay, Atif?
Yes.
Okay, done everyone? Am I audible, people? Yes, no, something?
Yes, sir.
Okay, very well. Just give me, all of you have NCRT with you? Just confirm. Yes or no? Rifat has, others? No, let me pull out this figure.
Yes, sir. I just brought.
So everyone has NCRT? Let me see which page you have to go. If everyone has NCRT, then for this part we can come to Come to page number 112, 112.
Yes.
Can you see table 6.1 there, people?
Yes, sir.
Yeah, just we give me one moment, okay? I'm just pulling out that figure. Yes, okay. So this part I will take. Okay, everyone. Am I audible? I hope I'm audible. You all can see the screen.
Yes, sir.
So I've taken this page from your NCRT, which basically you all can read, right? Is it clear to all for reading? Shall I zoom it more?
It's fine, sir. We have the NCRT.
We have been talking about this, the genetic code thing from a very long time, right? That there is a genetic code. So this is actually how the genetic code was decided. So basically, if you see in your table, how many amino acids are there? I told you
20.
20, right? 20 amino acids. Now, in this box, can you figure out the 20 amino acids? Let's write the name here. So I can see PHE, phenylalanine. Okay. PHE is for phenylalanine. Then, what is the next one you can see? LEU, leucine. Then, what else? What is different? Yes, ILE, isoleucine, right?
Yes, sir.
Okay. And MET, methionine. And what else can you see? You can see VAL.
Valine.
We have studied about VAL. What is it? Valine. And can you see that there is one GUG for valine? This is the one
Yes, sir.
that causes What does this GOG causes if it is present
Sickle
at
cell
the wrong
anemia.
spot? Sickle cell anemia, right? Very good. So apart from this, what is it? SER, which is serine, and PRO, proline, and PRO, THR, threonine. And what else?
Alanine.
Alanine, yes. Alanine. And what is there then? TYR. What is TYR? Tyrosine. Tyrosine is also, we have studied. Where we have studied? We have studied that if normally phenylalanine converts into tyrosine, right? Through an enzyme called phenylalanine hydroxylase, phenylketonuria. Remember, people?
Yes, sir.
Yes? Very good. Now, what else? Tyrosine is done. Where were we? Tyrosine. Do you see this UAA and UAG? These two? They are called stop. So these, they do not code for any protein. I'm just marking it in red here. Any amino acid, they are the stop codons. There must be one more stop codon apart from these two. And it's right here. See it? UGA. What I have marked in red are 3 stop codons. Apart from that, let's come back to the list. What is HIS? Histidine. And what is GLN? Glutamine. Then what is ASN? Any idea? It's called asparagine. Asparagine. Where have you heard this asparagine? I'm sure you must have heard of it. Try to figure out. What is LYS? These are the three letter codes. This is for lysine. And then LYS is done. What is ASP? It's called aspartic acid. Okay. ASN is asparagine or ASP is aspartic acid. GLU is glutamic acid. Where have we studied glutamic acid, people? This is what is the normal amino acid present at the 6th position that changes into valine, right? So that sickle cell anemia happens. Remember? Then
Yes, sir.
CYS is cysteine, 17th, cysteine. And then there's a stop codon. TRP is tryptophan, which is the 18th. Tryptophan. Then we have ARG. ARG is arginine. And then we have what is left? SER. SER we have done already, right? Serine. Okay. SER is serine only. This one. Then what we have? More ARG. GLY. What is GLY? Glycine. 20 done, right? So these are the 20 amino acids which are coded by the triplet codon. Write it all down. There are 2 more which has been discovered in animal system, but they are like some modifications of threonine. So there is one which is the modification of threonine, and I think one more. Now look here. So genetic code is basically, what is the concept here is, we only have 4 base pairs. Yes or no? Right?
Yes.
Everyone? And these 4 base pairs has somehow to code for these 20 amino acids. Now, how it can be done if you have only 4 combinations? So it was basically, there was a scientist called George Gamow. Okay. George Gamow was a physicist. He was not from biology. He was from physics, good in mathematics, permutation combinations. And George Gamow said, write down his name, is might be asked in entrance. George Gamow. Okay, this person argued that if you have in the DNA only 4 different possible things, because phosphate group is same, sugar is same, the only thing which is different in different nucleotides is the base. If you only have 4 bases and you have to code for 20 plus amino acids, you know, at minimum 20 acids, 20 amino acids, then what could be the only way? How long should be the code for one amino acid? Right? So he said that it has to be 4 raised to power, because there are 4 bases, raised to power 3, because 3 bases can together code for one amino acid, and we can call it a triplet codon, because 4 raised to power 3 is, what is the number?
60.
How many codons? Yes, 64 codons will generate, and this is the only one possible. So by this possibility, you can have more than 2 different codes for the same amino acid. Now look here, look at the first one. I'm talking about this. UUU and UUC. They both code for phenylalanine. Can you see this?
Yes, sir.
And from here till here, there are 6 different codons coding for leucine. 6, not 1, not 2, 6. Same as with arginine, you see, 1, 2, 3, 4, and 2 more. 6 for arginine. So there are 6 codes for arginine. And this, why is this happening? Is because, let's say, so DNA is very susceptible to errors and things like, so this is called degenerate. The code is degenerate. What does it means is, if you have UUU in the DNA and this was coding for a phenylalanine. Now, if somehow by mutation or in some other organism, this protein was very, very important and it changed back from UUU to UUC. Let's say there was a C that came, but still UUC will still code for phenylalanine. So you understood? Even if DNA has some subtle changes, it will not cause the organism to bear very big cost. Okay.
Yes, sir.
So there is some degeneracy. There is some area, gray zone allowed, where you can mutate the DNA a little bit, but still the protein structure, the protein function, and the amino acid will stay the same. The problem only happens in cases where glutamic acid. So let's come to glutamic acid, GLU, GAG. In this GAG, if there is any problem, it can become GUG. So if this GAG becomes GUG, right? So it's not like they both are coding for same amino acid. They're coding for different amino acid, right? So this is where
Yes.
disease will happen. You understood, everyone?
Yes.
Now, so this is the formula that gives how many different codes can be produced. So this box has 64 codes. How to read it? This is the first position, then the second position, and the third position. So if you take U from here, and from third position also you take U, so it becomes UUU, phenylalanine. If you take U from first position, U from second position, and C from the third position, it becomes UUC. You take anyone, this one. It took C for the first position, C for the second position, and U for the third position. Can you see this? How to read this square? Everyone clear?
Yes, sir.
Yeah. So this is the genetic code, and these are the salient features for it. So the first feature says the codon is triplet, as we figured out, because only a triplet codon can give us more than 20 plus combinations. And it also tells us that for one amino acid, there are more than one codes. So the codon is triplet, 61 codons code for amino acids and 3 codons code for stop codon. So there are 3, which is UAA, UAG, and UGA. These 3 ones, they do not code for any amino acid. If they are there, it will make the protein stop. So the protein, the length of the polypeptide chain will stop here. So this is important for you to know before we go to translation. Okay. Second thing is, some amino acids are coded by more than one codon. Okay. And therefore the code is called degenerate. It is a degenerate code. It's not specific, one code, one amino acid, not like that. The codon is read in mRNA in a contiguous fashion. There are no punctuations. It's in a continuous manner. The way we study it here, it's not like it will be UUU, AAGG, TTS, like that. Okay. The code is nearly universal. For example, from bacteria to human, UUU will code for phenylalanine. Some exceptions to this rule has been found in mitochondrial codons, and some in protozoans, but mostly it is universal. Okay. AUG, there is only one, and I'm marking it in green. Yeah. Have a look at this one single codon here. Can you see that AUG is the only codon that codes for methionine? Everyone can see the one that I'm marking in green?
Yes.
It has a dual function. It is also a start codon, and it also codes for methionine. So this amino acid methionine also acts as a start codon. It starts the reading frame of every protein, and every protein starts at methionine and ends at any of these 3. Is it getting clear, everyone?
Yes, sir.
So AUG has dual functions. It codes for methionine, and it also acts as an initiator codon or start codon that starts any protein in the world, in the planet, starts from AUG only. And then 6th, these three are terminator codons or stop codons. Okay, is it clear, everyone?
Yes.
Any doubts in this? So I'll end here at genetic code today, and in the next class I will start with translation, which is the last part in the central dogma. We have done replication, transcription. Today in the next class we'll do translation, and for that we have to read tRNA and also rRNA. Okay, ribosome and transfer RNA both are required for protein synthesis, and that will do. I think after that, what is left in your chapter? Can you tell me, Atif, just go down and tell me what is left. Operons will be left, operons, and I think what else? Human Genome Project. These 2 things are there. Apart from that, what else is there in the chapter?
Yes.
Operon and Human Genome Project, right? Basically, broadly. Okay, that's fine. That I'll see and I'll plan the next class accordingly. No problem. But is this clear? Today's class? Any question? Any doubt? Anyone wants to ask something? Everything is written. The amino acids. Everything understood with respect to the code. George Gamow gave this, argued this formula. He was a physicist. Just remember that. Okay, so I'll see you in the next class, people. Take care. Please keep revising and please get your mics fixed, Rifat and others. Please speak in the next class, okay? Don't make it just me and Atif discussing.